Hello, I am Mercè Boada, the founder and Chief Medical Officer of Ace Alzheimer Center, Barcelona. As a host, it's an absolute pleasure to introduce you to the first Global Research Summit. Since 1995, we've always had a clear purpose to bring Alzheimer diagnosis, treatment, research to a whole new level. Today, we are a renowned organization all over the world with a team of 100 professionals. Let me show you how we have got here. Since 1996, we have diagnosed around 30,000 people. We have collected 17,000 biological samples and led 138 clinical trials. Just now, as I am talking, we are doing 22 clinical trials targeting all phases of Alzheimer's disease and 9 national and international research projects, along with the highest level companies in the world. This allowed us to be heavily involved in the key scientific events of the last 2 decades. In fact, in 2020, even with the challenge of the pandemic, we have published more papers than ever before. Quite a journey, isn't it? But now it's time to set our sights on what's ahead with some passion and purpose from back in 1996. It's time to introduce you to the future of research. Welcome. Welcome to the Global Research Summit.
Good morning, good afternoon. I'm Marta Marqué, the Deputy Director of the Clinical Research Program at Ace Alzheimer Center, Barcelona. I'd like to welcome the audience to this first session of the Ace Global Research Summit 2021, co-organized by Ace Alzheimer Center and Grifols. As you know, today's session is going to focus on advances in molecular neuroimaging. We are very privileged to have a spectacular panel of speakers, including Dr. Victor Villemagne and Dr. Neil Basdev, who are going to talk about novel PET tracers for neuroinflammation. First, please allow me to say a few words about the organization of the webinar. Each speaker is allotted a 25-minute time slot. After the 2 talks, we will have 45 minutes for discussion and debate. Questions by the audience might be submitted through the chat during all the session and will be answered at the end during the discussion time. Although questions through the microphones are also allowed if you asked for it previously in the chat. You can also send your thoughts through social media. I would also like to take this opportunity to thank our partners for making this webinar possible and for their continued support. Lastly, I am delighted to introduce you to the chairwoman of the session, Professor Dr. Agneta Norberg. She's a professor in clinical neuroscience and director of the Center for Alzheimer Research at the Karolinska Institute, and senior consultant in geriatric medicine at the Karolinska University Hospital Halinge. Professor Norberg, please go ahead and start the session whenever you're ready.
So, good afternoon. I'm very honored to chair this session on a webinar on advances in molecular neuroimaging, and which is have a focus on and discussions about the role and the importance of glial cells and gliosis in different type of neurodegenerative disorders, and perhaps especially we focus on Alzheimer's disease. The first presenter is Dr. Victor Villemagne, and the title of his presentation is Astroglial Neuroimaging Markers in PET. And Professor Villemagne, he graduated cum laude in 1983 from the Universidad Nacional de Buenos Aires, Argentina, And he was awarded a postdoctoral fellowship and continued also with a postgraduate study in the Division of Nuclear Medicine at the Johns Hopkins Medical Institution in US. He subsequently also worked at NIDA and NIH and the University of Pittsburgh and did research. And after 2003, he joined the neurodegeneration group in Melbourne, and this was a very exciting time because it was also when amyloid PET PIB was presented internationally as a new tracer for visualizing of Alzheimer proteins in the brain, and he produced a lot of very interesting scientific studies, both on amyloid and later also on tau imaging. Very recently, in 1920, he left Australia and joined the Department of Psychiatry at the University of Pittsburgh. His research involves both national and international collaboration with neuroscientists, chemists, molecular biologists, clinical neurologists, and molecular imaging experts. And he has regularly attended and given presentation on numerous number of international meetings. He has authored 350 research publications and received several honors, including Monique de Leon Prize in Neuroimaging at AIC in Boston, US 2013. The Christopher Clark Award for the Continuation Advancement in the Field of Human Amyloid Imaging in Miami 2014, as well as the Colason Award by the Society of Nuclear Medicine and Molecular Imaging in Philadelphia 2018. Since 2016, he has been recognized as one of the world's most influential scientific minds based on his citation being in the top of 1% in the world in the field of neuroscience. So it's a great pleasure now to introduce Dr. Victor Villemagne, and again, the topic of his presentation is emerging PET astroglia neuroimaging markers with PET. Please.
Thank you, Professor Norberg, for the introduction. I would like also to thank Professor Boada for the invitation to present here. Here are my disclosures. The neuropathology of Alzheimer's disease is characterized by, as you know, by amyloid plaques and tau tangles, but they are surrounded by a neuroinflammatory reaction, mainly characterized by activated microglia and reactive astrocytes. Later on the disease, there's infiltration from the periphery of monocytes. They had been looking, several years, they had been looking at imaging and neuroinflammation, and they have been looking at different targets. The main one has been the translocator protein at the surface of mitochondria that gets overexpressed during microglial activation. But there are several other targets that are worth looking for. Professor Bassett will describe them for microglia. I will concentrate on astroglia this time. The main ones that have been used are MAOB targets and imidazoline 2 binding sites. As you look on top, there's imaging of microglial activation with this tracer PBR28 developed by in NIH by Professor Innes. As you can see, the astroglial activation increases as the disease progresses. So it's very high at the late stages of the disease. At the bottom, we see the carbon-11 deprenyl. This is our studies done by Professor Norberg at Karolinska. And here is much more interesting because if you look at top row here along the AD continuum, we have a high signal at the MCI stage with high amyloid. And even more interesting, if you look at the bottom, where you have the pre-symptom autosomal carriers of Alzheimer's disease, We see high signal in the pre-symptomatic carriers, but we don't see it on the symptomatic carriers. And reactive astrocytes have been said and even proposed as a hypothesis for Alzheimer's disease. And they are saying that they precede amyloid deposition in the brain, which is strange because being a reaction, you need to react to something. It might not be amyloid, but certainly you can detect it early on before having very high amyloid, insoluble amyloid deposits in the brain. Astrocytes are the most abundant cells in the brain, glial cells in the brain, and they are critical for the normal function and preservation of brain homeostasis. Among the many functions they have, they are involved in synaptic plasticity and formation of memory, regulation of the glutamatergic and GABAergic neurotransmission, regulation of blood flow, and contribute both to A beta production and A beta clearance. Reactive astrogliosis is a very heterogeneous reaction. This is a very simplified schematics of reactive astrocytosis. So essentially, you have initial macroglial activation that releases cytokines that lead to the reactive astrocytes, what's called A1 astrocytes, that have increased C3, increased GFAP, the glial fibrillar protein, and increased MAOB, and is involved in the release of neurotoxins that affect the neuron. It had also been proposed that the proteinopathies directly activate the resting astrocytes. And there's the A2 astrocyte that is supposed as protective, that is mainly triggered by ischemic events. The potential application of imaging is the accurate and early diagnosis of astrogliosis, the assessment of the spatial and temporal pattern of astrogliosis in relation to age, cognitive performance, disease progression, genotype, A beta and tau, and other disease biomarkers, and also used to validation of new imaging and fluid biomarkers for astrogliosis. And could be used also for disease staging, may be added as a module to the ATN framework. This early detection will allow prognosis and early disease interventions, and it can be used for trials for patient selection and monitor the effectiveness of the treatment. Most of the traces for astrogliosis have been concentrated in either imaging MAOB or imidazoline 2 binding site. The most used radiotracer has been by far carbon-11 L-deprenyl, and Professor Vaslet, who's going to be presenting next, developed another carbon-11 tracer, SL25. There had been much more recently proposed an imidazoline 2 binding site receptor, carbon-11 BU, 99008. And we have been testing a new tracer, an F18 derivative called SMBT1, and I will talk about more or less of the evaluation of this tracer. This tracer has very high affinity for MAOB, both in recombinant MAOB and AD brain homogeneous. The in vitro evaluation showed that when you correlate the amount of density of the bound SMBT1 to MAOB activity by radioluminescence, it has a very high correlation between the density of the binding in autoradiography and the activity of the enzyme, the regional distribution of the enzyme. There had been comparison with between SMBT1 and THK5351 that was originally proposed as a tau agent. As you can see, the structure is extremely similar. And I will talk later about THK. as you can see, when you do the autoradiography, the AD subjects present higher retention of both tracers. But when you complete with lavazimab, which is an MAO-B inhibitor, you wipe out all the signal from SMBT1, but there's some residual signal with the THK, and it's not competing with MK, which is a tau tracer, and PIB as an amyloid tracer. And here are the images in a healthy control, A beta negative control, and an A beta positive AD. As you can see, it has these are 60 minutes after the injection of the tracer. As you can see, very high activity in the brain, even at 60 minutes. And you can see in the controls the normal distribution, the regional distribution that is normal for the MAO-B in the brain, high in the brainstem and basal ganglia, and also in the mesial temporal lobe. But when you look at the AD, you see much higher retention in cortical areas like the posterior cingulate, frontal cortex, and essentially the temporoparietal cortices. And what is the usual criteria for evaluation of a new neuroimaging tracer? We look at saturable binding, binding selectivity, stereoselectivity, and regional brain distribution. When we look at binding selectivity, and we go back to the study with THK 5351, this is a study done in Montreal, and after a single dose of 10 milligrams legendine 1 hour before the scan, they show that they have a 36% reduction of the signal in cortex and a 52% reduction of the signal in basal ganglia, showing that this tracer is essentially an MAO-B tracer. We wanted to carry this further, so we did 5 milligrams legendine twice daily for 5 days. Here are SUV images showing in a control, amyloid imaging on the left, tau imaging in the middle, and before the selegiline and after the selegiline with THK 5301. As you can see, it wipes out completely all the signal in the brain. When we do the same with an AD patient, we have high tau in the brain, and after selegiline, it wipes very much of the signal, but there's some residual signal in the brain, probably attributable to tau, high tau in the brain. And when we quantify that, we see that in the basal ganglia we have about at 80% reduction of the signal. In cortical areas, we get about 72% reduction of 70% of reduction of the signal, and the AD block less than control, probably showing that they have this residual binding to tau. So we did the same with the new tracer, and we selected the 5 controls that were tau negative, and 4 AD patients that were high amyloid and high tau. Here are representative images of the SMBT1 on the left. And after the same 5 milligrams legendine twice daily for 5 days, we see that complete wipeout of very robust blockade of the signal, both in controls and AD. And in AD, we don't see any residual binding. And when we quantify, we see that we have more than 85% reduction in the signal in cortical areas and the cerebellum. And there's no difference between controls and AD. When we look at the kinetics and look at time activity curves, we see very high entry of the tracer into the brain, about 8% of the injected dose in the basal ganglia, and reversible kinetics. And when we plot the SUVR over time, we see that it reaches apparent steady state about 50 minutes after injection. So when we look then, follow up with regional brain distribution, and when we plot the activity, the regional activity of the brain with SMBT1 and compare it to the known distribution of MAOB in the brain, we see a high correlation between the two at more than an R-square of 0.8. And we also did it with different quantification techniques. And we find, for the tissue ratio, different reference regions. And we also found that a very high correlation between the semi-quantitative and quantitative measures and the regional distribution. We also look at the effect of age, and we compare amyloid negative controls against 10 young controls. And we see that all the regions in the elderly controls were significantly higher. So when we look at the frontal cortex, these are results with deprenyl than with PET, and we see an increase with age. This is with reported in vitro, again, in the frontal cortex, an increase with age. And we see the same increase with age with SMBT1. And here the images of these three females, amyloid negative controls, and you can see the red circles along the line where they show, and you see increased signal of SMBT1 in this control. So these are representative images, surface projection images of amyloid negative control. As you can see, no amyloid in the brain, no tau, but you see some signal with SMBT1 reflecting the normal distribution of MAOB in the brain. But when we look at that amyloid positive control, we see high amyloid in the brain, not tau, not much tau, except a little bit probably in the mesotemporal cortex. But we see that the amount of SMBT1 retention in the brain has increased and extending to more cortical areas like the orbitofrontal, posterior cingulate, and increasing and lateral temporal too, as well as the supramarginal gyrus. When we look at an MCI patient with high amyloid in the brain, high amyloid, now we have high tau, and even much higher retention of SMBT1 in more extensive and higher than before. And when we look at an AD patient with high amyloid in the brain, high tau, and again, an extensive retention of SMBT1, probably reflecting reactive astrogliosis. And when we reflect this, we display this as this column, we see that in the AD patients, we have high tracer retention, significantly higher retention in the posterior cingulate, superior parietal, supramarginal, gyrus angularis, and temporoccipital areas. But what's more interesting for us is that we also have significantly higher retention in high amyloid control and extending to the orbitofrontal regions. But again, the posterior cingulate, supramarginal, sun gyrus, lateral occipital gyrus angularis, which means that you can detect early on high retention of the tracer, probably reflecting early reactive astrocytosis. Interesting also, we found that in the AD group was significantly lower retention in the caudate, the same trend we saw it in the putamen and thalamus, and also in the hippocampus and parahippocampus. But we need to be more careful with that because this study had not been corrected for partial volume effect. When we do the correlation with amyloid and tau, we have a correlation with amyloid on top. We see high correlation in the temporoparietal regions, slightly less in the frontal region, reflecting very high correlation with amyloid. What is interesting is that not all regions with very high amyloid, like the superior temporal gyrus or the frontal, have very high correlation with amyloid, but other regions with not typically high amyloid in the brain, like the lateral occipital, has high correlation. And then when we do the correlation with tau, we have much less correlation, but again, in the supramarginal gyrus and a little bit in the frontal, dorsolateral prefrontal area. What was interesting is to look at the this correlation plotted as a graph, and when we do a fit, it is not very linear. Essentially, it seems that even in those who have subthreshold amyloid, the signal is increasing and then keeps increasing much slower when the amyloid becomes abnormal in the brain, suggesting that this astrogliosis might be a very early event in the AD continuum. When we correlate it with cognition, the retention is highly correlated with all the different tests and domains that we tested. But essentially, when we plot it, we enter into the model all the other covariants like age, sex, amyloid, and tau. We see that the main driver of the cognition is tau. And we have to remember that these are putting all the subjects, all the clinical groups together. So essentially, it doesn't look at cause, but the direction of the association. And why supramarginal gyrus? Why we mentioned that a lot? Essentially, because we find that it's high, significantly high in the three groups. But if we look at the correlation with amyloid and the regions that are significantly higher in amyloid positive controls, tend to be the same region with high correlation. And this looks more like a pattern that you see in glucose metabolism than with amyloid, or you see with tau. But these are areas with very early amyloid accumulation. As you can see here, the supramarginal, posterior cingulate, and temporoccipital. These are regions that even 10 to 15 years before reaching the threshold of abnormality already show some degree of tracer of amyloid deposition in the brain. And we're talking here insoluble amyloid, not soluble amyloid. Back in 2012, we published showing that what we call the temporoparietal junction and the precuneus were areas of the earliest amyloid accumulation, as you can see here in controls and MCI. And then in 2020, they published the same but with a different name, call it the banks of the superior temporal sulcus, showing that these are regions of very early amyloid deposition and also fast amyloid deposition in the brain. As a summary, reactive astrogliosis is a heterogeneous and early event along the AD continuum. Reactive astrogliosis is characterized by both a loss of protective function and a gain of neurotoxic function. Better and early characterization of astrogliosis may allow a more targeted or tailored therapeutic intervention, and this better characterization will allow a more accurate determination of prognosis, as well as patient selection and monitoring the effect of therapeutic trials. Conclusions in regards to SMBT1, it's a highly selective F18 MLB tracer with high entry into the brain and reversible binding kinetics that makes it an optimal PET tracer to be used as a surrogate marker for the in vivo assessment of reactive astrogliosis in clinical research. I would like to thank the people that made this presentation possible. Thank you.
It is now a great pleasure to introduce the next speaker, which is Dr. Neil Bastev. And the title of his presentation is Emerging PET Microglia Neuroimaging Markers. Dr. Neil Vastev is a director and chief radiochemist of the CMH Brain Health Imaging Center and also director of the Azrieli Center for Neuroradiochemistry at CMH. He's also the endowed Azrieli Chair in Brain and Behavior and a Tier 1 Canada Research Chair in Radiochemistry and Nuclear Medicine. In addition, he is also a full professor at the Department of Psychiatry at the University of Toronto. Dr. Vastev received his PhD in chemistry from McMaster University, and he continued training with a NSR postdoc fellowship at the E.U. Lawrence Berkeley National Laboratory in California. After that, he began his independent faculty career in Toronto in 2004, and he worked at CAMH for 7 years and was then recruited to Boston. So between 2011 and 2017, he served as the director of the radiochemistry at Massachusetts General Hospital, and he also joined the Faculty of Medicine in the Department of Radiology at Harvard Medical School, where he still maintains a faculty appointment. Dr. Vastev's research focuses on developing novel radiochemical methods and translating new PET imaging agents to use in various brain health illness. He has held continuous 3 Council and NIH funding and published more than 150 peer-reviewed articles and also delivered more than 100 lectures worldwide and has received numerous scholar awards throughout his career. He is also a fellow of the American Chemical Society, Royal Society of Chemistry, and Society of Nuclear Medicine and Molecular Imaging. So it's a great pleasure to give the word now to Dr. Neil Vastev. And again, the title is Microglia Neuroimaging Markers by PET. Please.
Thank you, Professor Nordberg, for the kind introduction. Today, I'll share our work on emerging PET radio tracers and targets for imaging neuroinflammation, and I'll focus on our outlook beyond TSPO. For those of you who are not familiar with our Brain Health Imaging Center, we're located within the University of Toronto's campus and have been in operation for over 30 years. We have over 40 PET radiopharmaceuticals for brain imaging. This facility has 2 cyclotrons, 17 hot cells, 12 which are for GMP productions, everything from medicinal to organic chemistry and preclinical imaging with 2 preclinical scanners. For human use, we have an HRRT and a PET-CT and a 3 Tesla MRI. One of the things that makes our center unique is that we offer non-traditional radiochemistry methods with carbon-11 and fluorine-18 to make tracers for human use. And we do a lot of work with the pharmaceutical sector for first-in-human, phase 0 and phase 1 studies. This slide summarizes the PET radio tracers we have for human use. As can be seen, nearly half of these were developed for first-in-human use, and most of the others were applied for first-in-human use in Canada at our site. These tracers cover receptors, transporters, and now we're focusing in on enzyme imaging and targets for neuroinflammation. These newer generation tracers often require developing new radiochemistry methods to make the new tracers for new targets, and that's going to be the focus of my talk. Now, I know this audience is very well aware that the most common PET radionuclides for neuroimaging are carbon-11, which has a 20-minute half-life, and fluorine-18, which has a 2-hour half-life. Because of the short half-lives, the vast majority of radio tracers are synthesized within 1 or 2-step reactions, usually from carbon-11 methyl iodide or from fluorine-18 labeled fluoride. A harsh reality is that most drugs cannot be labeled without changing the structure of a lead molecule because we have very limited radiochemistry methods. About 60 years ago, total synthesis revolutionized drug discovery by exploring multi-step reactions and providing the methods and reagents to synthesize virtually any drug. Our laboratory is exploring what we call total radiosynthesis including multi-step reactions. We've actually pushed the limits of carbon-11 out to 6 steps for human use and fluorine-18 out to 10 steps. Our ultimate but impossible goal is to label virtually any molecule for a PET. And just like most drugs fail, the other harsh reality we have to deal with is that most tracers also fail. So our goal is to label the molecules by whatever means necessary and test them in vivo as soon as possible. The question I like to propose for this meeting is, what if we could label any molecule for any target to study neuroinflammation? What would people be interested in? I'm hoping we can have some discussion around this topic. For the past 15 years or so, the answer I've been getting from physicians working in aging research is we need to develop more sensitive and early biomarkers of Alzheimer's disease and related dementias. We have relatively recently seen that the FDA has approved 3 amyloid PET imaging agents labeled with fluorine-18, namely Amyvid, Neurosec, and Vizamyl. While these are employed in seminal multi-center clinical trials, none of them are actually ideal radiopharmaceuticals for imaging early Alzheimer's disease. This figure shows a snapshot of the tracers we're developing for Alzheimer's disease and related dementias for receptor targets such as the muscarinic and glutamatergic receptors, as well as enzyme and intracellular targets such as tau, TSPO, monoamine oxidase B, glycogen synthase kinase 3, the endocannabinoid pathway, and we're also exploring the metal hypothesis of Alzheimer's. A lot of these tracers require novel chemistry methods to synthesize them, and I'll show a few examples of this work, starting on our development of tau PET tracers. So Siemens Molecular Imaging Biomarkers developed a new generation of tau PET tracers called T807, which was acquired by Avid and Eli Lilly and then later named AV1451 and then fluortaucipir. And actually a year ago, it was the first in-class tau PET tracer to be FDA approved. The original synthesis is shown here. It was semi-automated. It was a multi-step reaction and is not amenable for multi-center clinical trials and routine production. So our lab started collaborating with Siemens about a decade ago, and we patented a 2-step one-pot method that was fully automated and validated for human PET imaging. And this really gave us a head start while I was at MGH for early human PET trials in Alzheimer's disease and related dementias. The schematic diagram of our methodology is shown here, and as we know, thousands of subjects have been imaged with this tracer. Now this figure shows 4 subject scans with fluortaucipir, which is shown in the middle, and PIB, which is shown on the right, and the MMSE scores of these subjects is going down from amyloid negative, amyloid positive, mild cognitive impairment to Alzheimer's disease. We can clearly see the difference between the plaque load on the right from the PIB scans and the tau density, which is starting in the temporal neocortex in the subject with mild cognitive impairment, and this is spreading to cortical areas as seen in Alzheimer's disease. Now this figure shows some preliminary unpublished data with the same tracer from 2 American football players from the NFL. Both of these are shown at the top, and they're both symptomatic of traumatic brain injuries. The control subjects are shown below. The subjects at the top show activity in the cell side between the gray and the white matter, and that is consistent with postmortem studies. A second generation tau PET tracer was developed for Alzheimer's disease by Merck, known as MK6240, labeled with fluorine-18 and shown at the top. The synthesis used a semi-automated procedure with a modified liquid handler and stepwise microwave heating. And we were fortunate to collaborate with Merck early and to get funded with them through the Alzheimer's Drug Discovery Foundation and now the National Institute of Aging, which is still ongoing. We started to collaborate working on automating the radiochemistry for widespread use and to carry out imaging studies with this tracer. The image in the top shows a patient with Alzheimer's disease and a healthy control subject below, which has much less uptake in cortical regions. The tracer seems to be optimized for Alzheimer's disease, but not necessarily for non-Alzheimer's tauopathies. There are now several second-generation tau PET tracers under development by academic and pharmaceutical labs, and the landscape of the chemical diversity is shown here. Most are derived from fluorotaucipir, and the ongoing goal is to develop 3R and 4R tau selective compounds for imaging of non-Alzheimer's tauopathies. 4 repeat tauopathies were reviewed in PET by Rosler, Villamagna, and coworkers. And this year we reported on our work, which we attempted to make a 4R tau PET tracer with CBD solutions in Sweden in collaboration with the Mathis lab at Pittsburgh. The results of that study were just published a couple of months ago in the reference shown at the bottom. Now, the National Institute of Aging and the Alzheimer's Association, including several people who are in the session, have provided a research framework to create a biological definition of Alzheimer's disease. And these ATN criteria, which are now well known and stand for amyloid, tau, and neurodegeneration, have been established. These criteria are widely used in clinical trials, including in this recent report by Lilly on donanemab, which was published in the New England Journal of Nuclear Medicine, and used amyloid and tau PET biomarkers as readouts in their trial design. But there's still a real need for imaging biomarkers of neurodegeneration and neuroinflammation that go beyond TSPO. The peripheral benzodiazepine receptor, which was renamed the translocator protein 18 kilodalton, or TSPO, is the most common target for imaging in neuroinflammation. Our laboratories developed a second-generation tracer called Fluorine 18 labeled FEPA. We did some preclinical work in oncology and first in human translation for brain imaging in Toronto. TSPO has definitely been the mainstay of PET neuroinflammation research, but there are several limitations that require us to seek more sensitive biomarkers for neuroinflammatory processes. If anyone's interested, these TSPO and PET has been very much extensively reviewed by our labs and others, and the reference is shown at the bottom. So here is a snapshot of emerging targets for PET imaging of neuroinflammation that look beyond TSPO. We and others are working on some of these targets, including COX-1 and 2, monoamine oxidase B, P2X7, and also several kinase targets, which are really understudied by imaging biomarkers in the brain, but much more established in cancer imaging. While PET neuroimaging is established at quantification of receptors or transporter ligand interaction at the surface of the cell, it's much more challenging to access intracellular drug targets such as kinases. Cancer researchers have extensive knowledge of signal transduction pathways, including kinases, and I think the future of brain imaging can really learn from oncology. One kinase that we have a major interest in is glycogen synthase kinase 3. It's well known to be related to tau phosphorylation, and there's a GSK3 hypothesis of Alzheimer's disease. There's also many reasons to study GSK3 as a target of neuroinflammation, and some of the best preclinical evidence comes from a study showing in rat cortical glia that GSK3 beta isoform is expressed in both astrocytes and microglia and is activated following exposure to LPS, which is a very aggressive inflammation model in rodents. GSK3 beta has also been studied in the context of multiple sclerosis. Researchers have used in experimental autoimmune encephalomitis or EAE model, which overexpresses and found overexpression of GSK3 beta and increases disease severity. We were the first group to report on a GSK3 PET tracer back in 2004. This was over 15 years ago, and we have been continuing these efforts ever since. Our first attempt to image this target failed. You can see on the image on the left, there's no uptake in the brain of the rodent, whereas FDG lights up nicely like a light bulb. And this is after a lot of blood, sweat, and tears to make a tracer. And despite literature precedent, it didn't get into the brain. It really shows that as radiochemists and as we're trying to develop PET radio tracers for new targets, there are many, many places to fail. We can fail in the chemistry, might not be able to make the compounds. We could fail in the med chem by not finding potent and selective enough compounds to label. We can fail in radiochem by not being able to label a compound, but even after we surpass all those hurdles, we can often get stuck in the imaging where the tracer just fails to get in the brain or doesn't show adequate binding or specificity. I think studies like this are something we can also really learn from. It really shows that we need a high throughput approach to make radio tracers. We really can't be making them one compound at a time. In the pharmaceutical sector, they use combinatorial chemistry to do this, and this is something we really need to take advantage of in radiochemistry if we want to be making an impact in how we accelerate our first-in-human translation. So I'm going to show a few examples of some carbon-11 chemistry and how we're trying to transform the way we do things with C11 and mass production of radio tracers using library synthetic methods. Now, the most common carbon-11 reaction is methylation. About 15% of the drugs in the CNS have methyl groups. Generally, all carbon-11 reagents are derived from carbon dioxide, including methyl iodide and methyl triflate in general. But 75% of drugs in the CNS have carbonyl groups. So if we could be labeling carbonyl groups and using CO2 directly from the cyclotron rather than converting it to methyl iodide, we could really access much greater libraries of combinatorial libraries that are developed by pharma for different tracers for different targets. The question we had about 10 years ago is, can we use CO2 straight out of the cyclotron? And using these green chemistry methods like CO2 fixation, can we use it to make libraries of radio tracers? And it turns out, yes, we can. In the case of that compound I showed earlier called AR18, which was developed from Astra's pipeline that didn't get into the brain, we were able to use CO2 fixation. It was a very simple method. And green shows an amine, which represents the left half of the molecule. On blue shows another amine, which represents the right half of the molecule. We bubble in CO2 into a fixating base like BEMP or DBU. It's a very simple reaction. It can be done at room temperature. We just add POCl3 to dehydrate it and purify by HPLC. Very simple chemistry, no tricks, don't have to convert to methyl iodide, and we can make limitless libraries of unsymmetrical ureas or other scaffolds of carbon-11 compounds, quickly get them in vivo, and once we have a lead, we can then figure out how to optimize things later. To show this is not just an academic exercise, we've actually used this for a diverse series of compounds from ureas. We've also done libraries of carbamates. We've even made carboxylic acids and oxazolidinones. Most of this work is covered in our 2 reviews, and you can see the structural diversity that can be achieved here. We can also use carbon dioxide fixation to make fluorine 18 labeled tracers, which is another interesting approach, because you can imagine we can throw in any fluorinated amine, any fluorinated phenol to make the carbamate libraries, or any fluorinated amine and another amine to make limitless libraries of carbon-11 labeled ureas. Once we have these compounds, as I mentioned, we can take them in to in vivo studies. If we get a hit, we can then work on the fluorine 18 labeling. As I mentioned, we've been working on GSK3 now for about 15 years. Our big breakthrough came a few years ago when we started collaborating with Pfizer. We were able to crack their vault and find a drug that was a failed drug called PF367. Although it wasn't an ideal drug, it was an excellent radio tracer because this compound was very easy for us to label with methyl iodide, and it gets into the brain. It very quickly clears in nice reversible binding fashions, and we can show that it can also be blocked. So the selective binding and specificity was also shown with saturability as seen in this non-human primate scan here. This work is something that's also funded by the National Institute of Aging and is ongoing. Once we had a lead in hand, that compound called PF367, we then synthesized about 50 compounds, or actually over 50 compounds based on it. And we developed these to optimize brain uptake. We labeled over 6 of these, evaluated them in non-human primates, and now we have 4 lead compounds, including 2 fluorinated analogs that are under consideration for first-in-human studies. And we hope to translate one of these analogs to human use very soon. As can be seen in this scan, we got a 3-fold shift in increased brain uptake, and we're very excited about our leads that have higher brain penetration than the first generation. Another kinase targeting tracer that we are interested in is macrophage colony stimulating factor 1 receptor, also known as CSF1R. This is a receptor of tyrosine kinase. It's localized to the extracellular membrane of microglia and a small number of neurons, as well as peripheral macrophages. CSF1R signaling is hypothesized to activate and enlist macrophages at the sites of injury, and in Alzheimer's disease, CSF1R expression is increased. It's also been looked as a therapeutic for neurodegeneration and neuroinflammation, and seminal work was published by Johns Hopkins in PNAS a couple of years ago. In our lab, we were interested in that tracer, but before we delved on going to first in human, we manufactured the tritium labeled compound and did some in vitro studies, which we just published on within the last month. In our hands, the tritium labeled ligand unfortunately didn't show adequate selectivity as a tool radioligand, and we couldn't use it as an in vitro tool based on our imaging of rodent models of neuroinflammation. So we're no longer pursuing this tracer, but there's some exciting work happening with this tracer in human subjects by other labs. Another target we've been working on for a long time is monoamine oxidase B. MeOB is an established target for addictions and an emerging target in Alzheimer's disease and for neuroinflammation. The gold standard traditionally has been carbon-11 deuterium labeled deprenyl, which is an irreversibly binding radiotracer for this target, and was developed by Fowler, Volkow, and others from Brookhaven, and is a very well-established target, something that we've recently reviewed. Our goal was not to use deprenyl because deprenyl has a metabolite that's carbon-11 methamphetamine and binds irreversibly to the target. So we were looking for something that would be more reversibly binding. You can see we took advantage of CO2 fixation for an even easier reaction here. Here you have an amino alcohol-based precursor on the left, and you can just snap carbon dioxide right through with a fixating base and dehydrating with POCl3. It's a very simple chemistry, something we established about a decade ago, and we've advanced to first-in-human studies in Toronto. And this is something that we are now developing Fluorine 18 labeled derivatives in collaboration with GE and Karolinska, but extensive studies are underway at our site. Very recent news came out with this next generation tracer labeled with Fluorine 18 called SMBT1 for astrocytosis. Just was published in Journal of Nuclear Medicine. It shows a series of compounds and their structural diversity at the top. SMBT1 was derived from THK5351, as some of the earlier ligands on that scaffold had primarily binding to monoamine oxidases. So this was developed as a derivative of that. And Dr. Villamagna and co-workers also presented this in the International Conference on Alzheimer's and Parkinson's disease very recently. Here you can see a healthy control at the top, subject with Alzheimer's disease at the bottom with a much increased signal, and blocking studies shown on the right. So a very promising new F18 tracer for MeOB for astrocytosis. Another target we're very interested in is cyclooxygenases, COX-1 and COX-2. These are biomarkers of inflammation that have been targets of recent studies for neuroinflammation and neurodegenerative diseases. Briefly, phospholipidase converts membrane lipids to arachidonic acid, which is converted to prostaglandins by one of the two COX isoforms, COX-1 or 2. COX-1 is a constitutively active isoform, whereas COX-2 is an inducible isoform. Yeah, the work I'll show from our laboratory is very much focusing on both of these tracers with new tracers labeled with carbon-11. And this is done in collaboration with our colleagues at the National Institutes of Mental Health, Dr. Pike and Innes. Now, their lead compound is this compound here called PS13. PS13 is labeled with carbon-11. It is a highly potent and selective compound for the target with an IC50 of 1 nanomolar for COX-1 and selectivity over 1,000 nanomolar for COX-2. This is a PET scan from a healthy volunteer where we can see high accumulation of PS13 in the hippocampus. We're also working on COX2 in collaboration with the NIMH. Here's another tracer called MC1 for COX2, again, very potent and selective. Here we're seeing the radioligand tested in non-human primates, which have been injected intracranially with lipopolysaccharide or LPS to induce an inflammatory response. So this is the LPS model of neuroinflammation in non-human primates shown on the right. And you can clearly see the radioactivity in the brain prior to the injection, in fact, showing effective brain uptake and retention. MC1 has recently moved into humans for neuropet imaging and clinical trials as well. Recently, our laboratory took these two tracers and evaluated them in oncology. We're trying to study the differences between oncology and brain imaging. And you can see that PS13 is a promising imaging agent for imaging COX1. You can see the uptake in the tumor for ovarian cancer xenografts and nice blocking on the right. Our paper is in press in Journal of Nuclear Medicine, if anyone's interested in the current issue. Now, although many advances have been made in the field of neuroinflammation imaging, current radio tracers all target these pro-inflammatory or M1 phenotypes of activated microglia, but there's also a major interest in looking at the anti-inflammatory or M2 phenotypes. This is very nicely described in this review article by collaboration between Amsterdam and University of Pennsylvania. You can see the targets that are being looked at include P2X7, COX2, CB2, and TSPO, but there's also an interest in looking at these M2 compounds with P2Y12R receptors. If anyone is interested in reviews on the endocannabinoid pathway, we've certainly extensively covered CB2 as well as FA and CB1 receptors and another enzyme called MAGL. And the last topic I'm going to talk about is mGluR5, which is an exploratory target right now. There are 3 papers that I've just shown here. For example, my colleagues at MGH from the Brownell laboratory were working on looking at mGluR5 in ALS mouse models and inflammation in there. After that, in Scientific Reports, there's another publication a couple years later, so this was about 3 years ago, showing mGluR5 in CTE and traumatic brain injury mouse models of mice. And very recently, this year, we've seen mGluR5 inhibiting alpha-synuclein-induced microglial inflammation, which has impacts in Parkinson's disease. So the last topic I mentioned I'll talk about is some new chemistry. Our laboratory is focused on Fluorine 18 methodology. We and others have been really trying to push this long-standing goal in the field, which is to label non-activated aromatic rings with fluoride, and that's among the biggest challenges in radiochemistry. This has been extensively reviewed by us and others, and the references shown below. Our laboratory developed iodonium ylide radiofluorination to solve this problem and translated this method for human use. We checked a lot of our boxes, which means that it's a one-step reaction, it's metal-free, it's regioselective and compatible with a wide variety of functional groups. It's a very effective way to make labeled compounds, as seen in our papers, but also for drug development and radiopharmaceuticals. This is an example of a mGluR5 tracer called FPEB, which was typically synthesized in very low yields because of the non-activated aromatic ring. Using our new chemistry, we can get a 10-fold higher yield and a 3-fold increase in specific activity using this method. Because Alzheimer's disease is a complicated disorder with multi-tracers are often needed, here's a study showing PIB for amyloid, FDG for glucose metabolism, MRI for hyperactivity in the hippocampus, and whereas most neurodegenerative diseases show a decline, mGluR5 was upregulated in early mild cognitive impairment. So in summary, we're pursuing over 10 new approaches for imaging neuroinflammation, really looking at signal transduction pathways, kinases like GSK3, enzymes like MAOB, COX1 and 2, P2X7, P2Y12R, and ROS and mGluR5 are among some of the emerging targets. I think new chemistry is really going to drive the new tracers for neuroinflammation. These are those reactions such as iodine amyloid chemistry with fluoride, CO2 fixation to really expand the radio tracer toolbox. And numerous academic and pharma partnerships are underway and will be central for our success. Just wanted to thank all our wonderful funding agencies, the organizers, past and present, trainees and colleagues in our centers, and thank you all for your kind attention.
Yeah, thank you very much, Professor Bastek, and thank you very much, Professor Villemagne, for your extremely interesting and elegant presentations. And the floor is now open for the audience to send in questions or to ask questions to the presenters. I haven't seen any questions yet, but perhaps it's coming. Yes, we have something here. There is a question to Dr. Victor Villemagne here about SMBT1, and that is about the results on MCI and AD. I wonder, there was a very short comments about it, if it was differences between those. Can you extend a little bit more? What is the difference there?
We didn't see, essentially we only have 3 amyloid positive MCI, so we don't talk about much of the MCI. The ADs were slightly higher than the amyloid positive controls, but this is the first run, so we need more and more increase the sample size, because what Professor Norberg have shown is much more interesting. He's saying this early increase and then it drops. So we need, first of all, do more studies and then do longitudinal studies that will take care of seeing how it changes over time.
Yeah, I wonder a little bit. We are now in the corona pandemic. We are used now to hear about the first wave and the second wave and the third wave. And I wonder about the astrocytes. I mean, are there more than one wave? I mean, are there different peaks during the development? It's a quite long, if you consider Alzheimer in comparison to many other neurodegenerative disorders. I mean, you can have the disease when you detect it early, it can be 10 years, so it's a quite long time. And how do you think that the astrocytes is falling in? How do you consider the role of the astrocytes or the astrogliosis?
Well, astrocytes, when we talk about astrocytes, we think that they're all the same. They're extremely heterogeneous ones, and they react completely different. But essentially what it seems to be, and at least following what Professor Cuello in Canada has proposed, this is a first wave that is mainly astrocytes, and then a late wave that is mainly microglia. But we haven't done, these have been proposed, but we don't have the idea is to grab this group of controls, follow them up on time, and see how they changes over time. I don't think that is, even when we plot the amyloid versus the SMBT1, you see that after reaching a certain level, they don't go up, they go up, but much slower than at the early pre-high amyloid phases.
But could that indicate that your tracer is binding to a subpopulation of the astrocytes? So I'm sure that there are 15, probably approximately 15 different types of astrocytes. So,
yeah, definitely.
When
you have a reaction, you have the resting, some of them remain resting, then reactive. Some of them, or the majority of them, they will express GFAP, the glial fibrillary protein, and then some of them will express MLB, overexpress MLB. And the ones expressing MLB are the ones close to the plaque, around the plaque. If you have reactive astrocytes that overexpress GFAP, but they're far from plaques, they usually don't have the MLB. So essentially, you look really at the subpopulation that has the MLB.
Yeah, we know, for example, that GFAP, which hasn't been mentioned here, is also something that the pathologists are using for detecting astrocytes. And now it's very popular to measure it in plasma. But in brain, I think it's 30% of all astrocytes it's labeled. So that is...
For example, we compared the SMBT1 with plasma GFAP, and it's highly correlated. But they are both driven by amyloid. So, but we know that they correlate. There's, as far as I know, there are no overexpression MAOB without GFAP, but you can have GFAP ones that do not have the MAOB. And as you said, the reaction is extremely heterogeneous, and not everybody reacts the same way.
No, and then perhaps it's interesting to compare different traces because they might bind a little bit different. I think we had another question, but it disappears from. Can I get it sent to me again here, because it disappeared, so I couldn't read it. There is a question by Zhuang Tung. I think since I cannot read it, I ask Dr. Tung to make the comment himself, I think. Because I just see here that in the chat here that he is asking a question, but the problem with monoamine oxidase B tracer for astrocytes is, and then it disappeared here. I cannot see it. Please send it in some other way to me, and I can read the. If not Dong Tong can. Oh. So your compound is a pure monoamine oxidase inhibitor. I think that probably was the little bit of the question here. I mean, can we think that there could be other?
I don't think it binds to anything. It's very selective for MLB, and the advantage also, it has very low non-specific binding. So yeah. The advantage with this tracer, we can do things that we cannot do with amyloid or tau. Given that the amount of binding time we have in the brain, we cannot compete with anything because it's really toxic. But with like receptors or enzymes, we can compete and find out if they're selective or not. And the other thing is that there was no residual activity when we did compared to the THK, which are kind of similar scaffold, similar compound.
There is also a comment by Dr. Savier. Then he also disappeared here. I'm sorry. I think I have to.
Dr. Savier, yes.
I have to have the question in another way because they just disappear from my screen when I see it.
Yeah. It was Dr. Sevigny, if it's the same one, I think, is the one who developed the aducanumab.
Yeah. We are talking about the microglia and astroactivation and astrogliosis. This is a question for the two speakers. I mean, do they have different properties? Are they working together? And you mentioned here that there are speculation that they are in different phases of the disease. What opinion do you have about these two?
Okay, let me go first. I think that they are working together. Essentially, is the early initial, even though you don't have the overexpression of TSPO, this early microglial activation releases the cytokines that leads to reactive astrogliosis. And they fulfill, in some aspects, similar but complementary functions. So macroglia will turn into macrophage and will try to get rid of, in the case of Alzheimer's, of amyloid. There's a small phagocytosis also from astroglia, but they work together. The problem with this is when this process gets chronic, so they start losing the protective function, and they start gaining the neurotoxic function. Neil.
Yeah, I completely agree with Dr. Villamagna there. This is a complicated process, and we don't have an established neuroinflammatory marker like we do for a lot of the other targets, right? If we're looking at, for example, dopamine transporter or something like that, it's very easy to say, okay, this is the gold standard, but neuroinflammatory processes are complicated, and we need many tracers, we need many studies. This is going to be an ongoing battle to translate as many tracers as possible to look at as many different pathways.
The COX is more related, but most of the other ones are surrogate markers.
Absolutely, I agree. And that's another thing. If we're looking at almost indirect measures of neuroinflammation and then basing it on our hypotheses, we need very large N numbers in our studies to draw any solid conclusions. What's nice, though, is we do have more animal models. We have post-mortem studies that can help guide us, and there's been a lot of literature in that space. But I think if for in vivo PET, we're really at an early stage, just at the beginning stages of unveiling what's going to be there.
The problem with neuroinflammation is the last 30 years, we have been probably in the same place. Since we started with PK, we haven't.
Yeah.
We can even trace it with arachidonic acid, but we never. The COX, the COVID group was following that probably 10, 15 years ago. They dropped it. Now INIS dropped the TSPO and picked the COX. So it's a very complicated thing. It's not. Let me put it, if you're doing the studies, the results are never very satisfactory. Yeah. So.
We have now a question from Dr. Xavier Morato. It would be also nice to correlate brain imaging as gastrogliosis microglia activity in early future with cognitive evaluation in relation to Alzheimer treatment. How can we calculate the maximum recommended safety, maximum exposure to PET tracers? So here is a little bit of several PET tracers, what I understand, at the same time.
But for example, well, you're talking about the radiation exposure. Okay? The advantage of these tracers, and Professor Norman, you were one of the first ones to start doing this, is that you can use the first phase of the injection of these tracers to get that kind of pseudo blood flow FDG image. So you have an idea of blood flow metabolism and the specific target you're measuring. So in a way, you simplify, you get two studies with the radiation dose of the other one. And essentially you need, if we're talking Alzheimer's, we're talking, you'll need the amyloid, especially when you're looking at early phase, and then looking at your specific inflammatory tracer, either microglia or astroglia.
I think it will be needed if you get this immunization therapy, that you probably have an amyloid PET scan as an inclusion criteria, and then you probably want to follow up and see if there is a reduction.
Yes, but this is something that we're talking. You need really quantification, because you don't know what happens with the treatment.
Definitely. Yeah.
And the advantage with that is that if you do real quantification, you can get R1 parametric images that actually are reflecting blood flow.
We had another question here from Dr. Ree Ask. If astrogliosis might occur in advance of overt protein deposition, I wonder I want to know which molecule signal are boosting this phenotype.
Well, the problem is that we don't know. When we look at imaging, in, for example, amyloid, we're looking at insoluble amyloid. And to get the signal for PET, you need to build certain amount of density of binding sites to get the signal. For astrogliosis, you might see it even earlier. And this is the study with Depranyl that Professor Norberg has published, and showing that you might even see it very early on, even though when you don't have too much amyloid in the brain. But you have to remember that there's other kinds of amyloid, like soluble amyloid, that we don't measure with imaging. And it's soluble, it doesn't last too much long in solution, probably 2 hours, and it aggregates. But that might be what triggers the astrocytic reaction. We don't know.
No.
Yeah, the point on soluble amyloid is interesting too, because I think isn't the Vmax for that would assumingly be very low. So can we access it by PET?
No, because it's less than 1% of all the amyloid in the brain, and it doesn't last long. They have been trying to do these antibodies, but essentially the antibodies go very little into the brain. They usually label with, you have to label with a very long isotope, but even the soluble doesn't remain soluble very long. Right. So it's not that you have a target, you have a target that is disappearing.
Yeah, can't access it, which is the big problem. Can't access it, can't drug it, can't image it.
Yeah. Well, actually, if you look at the donanemab studies, I think you for antibody for treatment, you don't need that much. For imaging, you need a lot. You need at least 5% of the injected dose in the first 2 or 5 minutes. But for treatment, you don't need that, essentially because you keep it circulating, and even if it doesn't go into the brain, it does. The problem, we are treating Alzheimer's very late, where amyloid is not doing much.
Yes.
We had another question here. Then let's going back to monoamine oxidase again. And the question was, the problem with monoamine oxidase B tracers for astrocytes is its non-specificity. Can you comment on that?
Yes, all the traces we have for neuroinflammation are non-specific. That's why we had to make sure that we mention these are surrogate markers. The other thing that we can be more confident is that regions that are high on control, the normal distribution of MAO-B in the brain, do not go up with Alzheimer's. So we have other regions, cortical regions, suddenly showing up in Alzheimer's. That with people the same age match that do not have Alzheimer's, those regions do not show up. But I would say that, as I mentioned before, probably with the exception of COX, that we're measuring the enzyme directly involving in degrading this arachidonic acid. We usually, all the TSPO and everything, they're all surrogate markers. There are no specific markers. So the MAO-B, you also have MAO-B in neurons, much less. When you look at TSPO, you also have TSPO in glia, in astroglia, but much less. So we do not have something that is targeting. As Neil mentioned, probably the purinergic receptors that tend to regulate microglial activation, and they get upregulated in the surface, maybe is closer. But again, it's not, like Neil said, it's not looking at dopamine receptors directly. So
Dr. Basti mentioned in the presentation the 8N criteria, and it would be interesting to hear from both of you the total tau, which is a part of the 18 criteria measured in CSF. It has been discussed whether it's a good marker or not, and discussion whether it should be replaced by something. So what do you think about the inflammation marker that has been discussed? And which inflammation?
I can go first. The total tau in CSF is not very good. NFL, the neurofilament light, performs better both in CSF and plasma. For neuroinflammatory markers, there's a lot of cytokines that you can measure in CSF or plasma that are usually released during the inflammatory process. But for the ATN classification, I wouldn't, for the N, that had been initially proposed total tau, I would use the NFL and CSF. I would use the glucose metabolism, or I use the MRI. That's why it's put in brackets, because it's a non-specific marker of neurodegeneration.
But what is your, what is then your opinion about the discussion about GFAP and plasma, that you can use that for measuring inflammation, and you can even hear the comments that you will not need imaging. You can just take. What is your comment about that, as pet persons?
Yes, the problem is that you have to be honest too. So with amyloid, when we started, we were talking to Bleno, Zetterberg, saying, okay, you spend all this time and you can only give one number. We see the whole brain, and after 15, 18 years, we give them one number, but we have the only thing that they cannot have. We have colors. So the plasma, A beta, might be a very good way of screening patients. The GFAP is very good to detect inflammation, let's put it this way, but it's even better in plasma. GFAP in plasma is better than CSF, but GFAP in plasma is better than the A beta 40 to 42 ratio, or 42-40 ratio. In plasma, we see more to predict amyloid. It has a much better separation.
I suppose
the whole reaction is driven by amyloid.
I mean, if the aim is to take a plasma sample for diagnosis diseases like Alzheimer and telling patients what you have found, I must say that it's much more difficult if you have the patient and the relative in front of you, and you said that, yes, we took a blood sample here, plasma, and the value was high or low. And they look at,
I would
don't understand. But if you have, as we do, we have the scans in our computer. So we just show in the computer and tell them that, look here, you see here it's amyloid all over the brain. That's explain your problem. Or look upon the glucose metabolism. It's here on the left side in the parietal cortex. And that's why you have some problem with your orientation to orientate, for example. That is more, then one really can explain the underlying mechanism, or what is the...
I totally agree. I think that we should have these whole panels of plasma,
yes,
that will give us an idea, okay, this person should have an amyloid scan. And actually then follow them up with FTG. People don't use FTG, don't want to use, they want to play with the new toys. But FTG is extremely useful, because it's measuring function. This is what you say, it's here, it's progressing. We don't have the same with amyloid. We might have a little better with tau, but tau is earlier in the sense of how it progresses, translating into cognition. So
we have another question here.
Just to follow up on that point, I think we have to think about it in the context there as well. I mean, I understand that it's expensive to do PET. It's prohibitive in a large scale. You're not going to put 3 people in person into a scanner for 3 scans, 1 for amyloid, 1 for tau, and 1 for neurodegeneration. Once we have an established neuroinflammatory marker, we're not going to do that around the world, but we're getting a lot of information from these studies. And I think where we're going to see the impact of the imaging and from the perspective of a PET person is going to be in the drug development. We can start looking at target engagement of new drugs and patient selection for trials. We can also figure out dosing regimens. We can really use the power of PET to drive the therapeutics, which I think is the ultimate goal. And that's why I wouldn't dismiss the imaging work that's being done. It's still at a very basic science stage right now relative to what we're trying to do for the masses. But I think it will continue to have impact. So I think one of the questions was frame it in the context of an imaging person. And sort of that would be my argument for why I think our efforts to pursue these goals are still going to be valuable.
We also have another question here, and that is a little bit about if neuroinflammation with PET tracers and studies has that been performed in non-AD dementia. What are your comments about that?
Neil, do you want to go with that?
Yeah, I think it's both microglia and astrogliosis. Perhaps
the most typical would be MS, but Neil. Sorry.
Yeah, those were definitely, we and others are obviously, several labs are looking at many different, more selective agents than we have right now. I mean, for MS, there's a whole series, for example, of Bruton's tyrosine kinase inhibitors that are being used as therapeutics, and we're making imaging diagnostics along there as well. In terms of non-Alzheimer's dementias, we're looking very intricately right now at, for example, PSP, progressive supranuclear palsy, which is mostly a 4R tauopathy, but then you've got Pick's disease, you've got FTD, you've got other DLB, you've got things that are also considered 3R tauopathies, and then CTE, which is a mixed 3R, 4R tauopathy. So a lot of work has been done with these. For example, with TSPO being the most widely used ligand, I'd say the bulk of the work has been done with TSPO and or FDG, and also with different generations of tau tracers. The first generation tau tracers seem to be a little bit better in a sense, like Merck's tau tracer is very good for Alzheimer's dementia, but not so selective for non-Alzheimer's tauopathies, whereas in non-AD tauopathies, there's tracers like PBB3 or Apranoia's APN1607, which has been renamed. That's considered to be a marker. Life Molecular Imaging has 2 tracers in their pipeline. I think one of them is PI2620 and then PI2014, again, looking for 4R tauopathies in those non-Alzheimer's dementias. But I think these new neuroinflammatory markers will play pivotal roles, especially we, and I'm assuming Victor's group, he can comment on this, we're all kind of betting right now on MAOB.
The main thing is that the non-AD dementias, all of them have an inflammatory component. ALS, frontotemporal dementia, even in the Creutzfeldt-Jakob. So they might not have involvement of amyloid or tau, but they have a neuroinflammatory component. And because the term neuroinflammatory says a lot, but it says nothing, we use the tag, but most of the studies, as Neil said, they were focused on looking at microglia, essentially because most of the time what we have, we're looking at TSPO tracers for the last 30 years. So. But the study that had the non-AD study that mostly looked at neuroinflammation, it was a goal, it was MS, trying to predict the flares of MS.
But there is also new studies coming for the astrogliosis in different non-AD, and I personally think that it's very interesting, and we are doing some studies to compare also different type of non-AD dementia with AD, because I think that even if there is an astrogliosis, it might be in different stages of the disease. So it seems, for example, that in frontotemporal dementia of the behavior variant, you can see a very strong strong astrogliosis when you have a clear disease. It was also a publication just the other day published for microglia in semantic dementia with quite clear microgliosis. So while in Alzheimer probably it's earlier and not so strong. So I mean there are different time courses probably also related to different pathological mechanisms. So that's I think you have to.
You published studies on Grifols Jacob with carbon 11.
Yeah, that's a definite very strong. Have you seen?
Yeah, I've seen the images. Yes.
You have seen the in cerebellum. The astrogliosis is in cerebellum. It's extremely strong.
Yeah. But one of the most interesting ones would be semantic dementia.
Yeah.
Because semantic dementia, some of these tau traces that are supposed to be 3 and 4 repeat binds to the temporal pole, but it's not to the cortex, essentially because there's not much cortex left. It might be to the astrogliopathy, like RTAC. So that's the other thing. And then looking on another TDP43 is what now they call late, the hippocampal sclerosis also. So I think that we have been looking at microglia all these years. It's because we have the tracer. It's not that because we thought that only astroglia was running the show. I think they are both important, and it's the interplay between the two.
Yes, I ask the audience here, send a little bit more questions. We haven't got any more just now. I want to ask you a couple, both of you, one question, and that is a comparison to amyloid. When we did the first PIB study, in fact, in Uppsala, and we were standing just at the PET screen and looked upon the uptake of the tracers, and we looked upon different regions, and we were very surprised, and we hadn't expected that there should be so high uptake in the frontal cortex, because I mean that is a region which is affected. In this case, it was a patient that was had a mean mental of 27, so she was quite early in a stage of Alzheimer. So I wonder regarding now astrogliosis and microglia activation, is there something that you have been surprised when you have done these studies and what unexpected?
Yes, because if you compare to your FTG, the frontal is very late.
Yeah.
And with amyloid, the amyloid is very early.
Yeah, but why? Why do you have a lot of plaques in the frontal cortex?
But what we have is there's an upregulation of choline acetyltransferase. So in a way, we have a way of compensating the amount of plaques there in the frontal cortex, which in MCI goes down in the posterior cingulate. So I think it's not mainly the neuroinflammatory process, but it's all the other
unexpected in the monoamine oxidase inhibitor tracer or in the TPSO tracers. I mean that you.
Well, for us, when we did the correlation, even though the frontal one has the region of higher amyloid, it didn't have the highest correlation. The same with superior temporal. Superior temporal has a lot of amyloid, not much. Anterior cingulate is also a normal distribution of MAOB. It doesn't increase much. So it's always something else going on.
So what do you think will be next step? I mean, are you not surprised that no studies has been done with this immunization therapy, which has been ongoing for so long time now? I must say that there is an activation of microglia, but and man has seen it in animals, but there is no data in the immunized patients, as far as I know.
They haven't looked at that.
No.
And I think they already have. The studies are very expensive already, looking just at the amyloid. I don't know if they will probably, if the study have CSF, they can go back and look at the inflammatory markers.
Oh yeah, but that's another.
That's for example,
Yeah.
that's one thing that we can propose to Merced.
Yeah.
All the samples that CSF sample look at inflammatory markers.
Yes, of course.
Right, Marta?
Right.
Yeah.
There's a new question, I think.
Yeah, we have a question here from Don Juan Logarta. Ask, is there any functional imaging data on resolution of brain-human inflammation, autoimmune infections when treated by steroids, immunosuppressants, or antimicrobials?
I don't know. The one thing that you have to be careful with looking at MAO is smoking. Smoking reduces MAOB. So you cannot do studies in current smokers. But then we try to avoid doing study with people that are undergoing heavy immunosuppressants because it's and try to avoid steroids too.
Yeah. Have you seen any differences between early and late onset regarding astrogliosis and
we don't have enough yet to look at that. What we see is the increase of in people with no amyloid, the increase with age. Yes, as was reported, but we didn't. And actually, that. Yes, we see the increase with age.
In postmortem brain, I mean, I think it's interesting also to analyze findings in postmortem brain tissue, because that is really the final stage of the disease. With PET, you never can image a patient when they are really in the advanced stage of the disease.
Yeah.
So there you can see a higher astrogliosis in old Alzheimer.
Yeah.
So it goes, and that is, I think it's different from tau, where you have more tau in the early onset. We can see if we have some more. Probably it's coming some question more here. It takes some time here, I think. Yes, we have something here from Dr. Javi Olarzan, who asked, could it be that specific molecules like TREAM2, SOX, are more relevant for Alzheimer pathology than glial cell type? Any way of tracing these molecules?
There are quite a bit of efforts ongoing for TREM, being led by Michelle James's laboratory at Stanford, and there's also a lot of industry collaborations. I know other academic sites are also working on it. I think it's pretty early stage, and it's interesting as an avenue. I think historically one of the challenges uses generally the selective molecules tend to be quite large molecules, so getting adequate brain penetration and everything is a challenge in developing these tracers, but there's certainly some nice work underway already by several labs. I think TREM is definitely an up and rising target, or something worth considering. And just to come back to Dr. Nordberg, to your earlier point, I think the field really needs to do much more of the basic science. And we spoke about this on the pre-call with you and Dr. Villamagna about this, where I think we have to do a lot more of these fundamental science studies. That includes the autoradiography that you just mentioned. That's the binding assays, the classic work with tritium ligands, to really try to understand the mechanisms before we delve too deep into the imaging or commit to a tracer. A lot of these de-risking studies really pay off, and they historically have. So it seems to be a bit of a lost art that I'm hoping will revamp over time. And the other thing too that we were discussing earlier, which is probably worth bringing up for discussion at the stage, is doing more quantitative analysis. It's another thing that we were discussing earlier that I think is worth discussing or bringing up with the audience, that we really have to do more rigorous studies for radiometabolite analysis and arterial input functions, and really try to understand the tracer kinetic modeling. This is another field that's we're losing a lot of the skill sets as we advance, which should be the opposite. I feel like we should be investing more into the fundamental science, not less.
People like pictures, colors. Yeah.
Yeah, but I think I totally agree here that the in vitro studies reveal that it is very complex. When we look upon different tracers, they are not perhaps binding just to one site. If you consider, for example, tau or amyloid, especially tau, and you can from in silico calculation and from cryo-EM studies now see that there are different binding sites on the tau fibrils, for example, for astrogliosis and microglia. Perhaps we don't know so much yet, but certainly there are interactions and different traces can bind differently, and it can be involved in different way in different diseases. And this is a little bit difficult to perform in vivo in patients. I mean, Victor, you're working with blocking. It's a general blocking of the whole monoamine oxidase, but it is. I think we have. It's a complementary. I think we have to have that knowledge.
Yes.
Otherwise,
well,
I think
that's why I put the idea of how to evaluate a new tracer. That you need to assess that. The problem is that with amyloid and tau, we haven't done it because it was impossible to do it because of the amount of binding sites. But we had to go back to basics. And essentially, for example, a peptide like amyloid, you already showed that they might have two different binding sites. It's the frontal amyloid and the hippocampus.
Yeah.
Imagine a tau, which is much, much larger. And then now they have these cryo studies and showing where they bind. So
that's a really important point that you just made, is that with amyloid especially, or tau, you have the targets are of massive Bmax, and the ligands are in with KD values in the low nanomolar range. So our Bmax to KD ratios are absolutely massive, like over 1,000 compared to what we would have for traditional receptor ligand interactions, where you only need Bmax to KD ratio of about 4 to 10, for example. So, but yet we're only binding a small fraction of amyloid or a small fraction of tau, even though the targets are so massive. But I think, Victor, you really hit the nail on the head there with the cryo-EM studies showing that tracers bind to different sites within, for example, for our tau or something else. Like, I think things are getting more and more complicated as we go for these targets, and we have to start thinking about things a little bit differently, especially when we're going for these neuroinflammatory processes, which are very dynamic and challenging, and going, spanning astrocytosis through microglial activation. These are not straightforward targets, not the lock and key mechanism or the high Emax targets that we were looking at before with receptors and transporters.
Very heterogeneous targets. They're not all the microglia, not all the astrocytes are the same or respond the same. So,
what do you think about? There was some papers quite recently speculating that the tau was taking up astrocytes, and from there it was spreading.
I don't know.
In the brain.
Yes.
Is that an important interaction, do you think?
I think, based on what we're saying, is we might have a close association with tau, but it's the tau in dystrophic neurites. They're the ones around the plaques. And there's phagocytosis in astrocytes. Mentioned before, is not the main function, but they also do that. But I don't think that the propagation of tau is through astrocytes. Astrocytes is much more clearly related to amyloid clearance based on the food over the blood-brain barrier, aquaporin 4, production of E4 or ApoE. So, but for tau, I think, based on the studies of the Mandel curves, it seems that the kind of normal tau is released, and when it's uptake, it starts aggregating on the other cell. But the main things that they have been proposed is the pre-release for tau, the microchannels, and then exosomes. So
But we have a different location of astrogliosis and amyloid. I mean, if you look upon the cortex, the astrogliosis is only in the superficial, mainly in the superficial layers, and then deeper, while you have amyloid in the whole, and then the astrogliosis in hippocampus is in all layers. So what do you think? Is there a difference there in?
I don't know. At least looking for MLB.
Yeah.
The MLB are the astrocytes related to plaques. You might have astrogliosis further away, but they usually don't overexpress MLB.
So I have a question for both of you. I mean, it's a really extremely rapid development in this field of Alzheimer, and I mean, we can just look upon the plasma markers as we have discussed. They have turned up now within 1 year, and so quickly. And if we have discussed the tau, and I think you mentioned the first tau traces, and they came. 2013, I think, was the first one, and then it's quickly, and amyloid was a little bit longer. So if we now look 10 years ahead, how is the landscape, and how is the landscape in research, and how is it in the more clinical setting? Where do you think is there any room for this type of glial cell markers, both in the research and or is also in the clinical use?
I think it's going to be both. I really do. I think this is one of the most active areas of research right now. We're seeing so many labs pursuing radio tracer development and discovery beyond TSPO and translating these compounds rapidly for first-in-human studies and also for multi-center trials. So I think we're also seeing the same trends from pharmaceutical sectors with their drug development pipeline. So I think these ATN criteria are being treated very seriously in Alzheimer's disease. So really the race is on to come up with established neuroinflammatory, neurodegenerative biomarkers, especially with PET. That being said, it's also an open field in a sense that we could really try many things. As Victor and I described, like both our centers are running with tracers, and yours as well, Agneta, with MAOB, for example, as one target. But I think right now there's so many different tracers, so many different targets, and labs are looking at different ways to image these different targets. We're going to start seeing not only clinical research, but hopefully actual, beyond just the research angle of it, and actual tying these studies as readouts for various diseases, especially things like non-Alzheimer's tauopathies and other diseases.
NIH already established that the main focus, at least for the next 2 years, is going to be inflammation, at least for the NIH for.
That's good, and I think we also need money, and I personally hope that there has been a trend, I think, now to focus very much on these very large databases, and it is a little bit less expensive to download a lot of scans and don't do all the work of recruiting patients. But I hope that it will continue, that people really would like to do and recruit patients and do PET studies, and more specific PET studies than these very, very large cohorts from.
Oh, I remember about 7 years ago, the European Union tried to have a consensus about neuroinflammation, which tracer to use to gather collision. And then everybody ended up with a different tracer. So it didn't change much. I think that was the last initiative, big initiative, to fund the neuroinflammatory markets over there. Yeah.
But I think we can now go beyond TSPO too, because at the time, PK11195 would have been the standard, and everybody was getting into the second and third generation ligands. Now we're at a stage that people are looking beyond TSPO, and we can really start seeing new.
I did the first PK studies back in '89.
Hopkins.
And it was bad then. It's so bad it cannot even pick up the polymorphism. Yeah. So.
So I think we are now approaching end of this this webinar on astrogliosis and microglia in Alzheimer's disease. I think we agree here that it's a fascinating area and it's very active, and as was also mentioned here that it's one of those who really are in focus now. And we see during the recent year that more and more webinars and discussions about it, and I think we can look forward to a lot of results. And that's, I think, is very important, because I think still unfortunately, I mean, we have no cure for Alzheimer's disease, and we have to understand the disease much more, as well as also the other, as you have mentioned, non-AD tauopathies and proteinopathies. And I think this is stimulating for further research and also application into the clinic. So I think with this word, I want to end, and I will thank the speakers, and I will thank the audience, and I give the word to the host, please.
Thank you. So yeah, we have come to the end of this session, and again, I'd like to thank Dr. Villemagne and Dr. Basdeb for their very interesting talks about this developing topic. Obviously, Professor Norbert for chairing this session, our partners Grifols and Care Pharma, and all of you from the audience for the questions and the discussion. And I'd like to invite you to join us next month for the second session of ACE Global Research Summit 2021, which is going to be on May 28th, also on a Friday afternoon. That session will focus on molecular biomarkers of neurodegeneration and will be hosted by my colleague, Dr. Agustin Ruiz. And now I would ask you to please help us improve these sessions by taking just a few minutes to fill out the survey that will come up to your screen in a few seconds. So thanks again, everybody, for joining us today, and we hope to see you very soon again for another afternoon of research. And have a good weekend, everybody. Bye-bye.
Bye. Thank you.
Bye.
Bye-bye.